So we're very pleased to have with us today Dr. Bob Macdonald. And Bob is somebody I've known for many years. Bob is currently the professor and chairman of the Department of Neurology at Vanderbilt and a professor in pharmacology, molecular physiology, and biophysics. He did his a BS degree at MIT in electrical engineering, maybe why he's an epileptologist, I don't know. He spent some time in Ohio at Case Western, and then did a Ph.D. in physiology at the University of Virginia, and then medical school and neurology residency there. He was the chairman when I was at Vanderbilt for a number of years, and I really appreciated his the wealth of knowledge that he had, and I think he made a huge impact on me and my career. He's done a lot of things nationally. He was the Catsius Award winner and lecturer a number of years ago. I was there for that lecture. I probably heard 30 of those lectures over the years, and that was one of the most engaging and understandable of those lectures that I think I've ever attended. So Bob's research interest has been in basic mechanisms of epilepsy, particularly related to GABA receptors, and we're so pleased to have him with us today.
Well, thanks again. Thank you very much for inviting me to join you in this first neuroscience symposium, which I'm sure is the first of many. I also want to congratulate you on the progress you're making to developing the Neuroscience Institute. I agree neuroscience is an extremely good area to grow and very important. It's also fun to catch up with Ken and Brian Ludwig, who has also spent time at Vanderbilt, to see both of them are prospering and doing extremely well. So let me get my talk queued up here. Or not? So you talked, so far we've heard a lot of talk about translational research, so whether you like it or not, I'm going to do it to you. I'm going to talk about a project that I think is quite translational, and that is dealing with the genetic epilepsies and GABAA receptor mutations. This is an area that in my lifetime has exploded. Back when I was a resident, it was not clear that a certain number of the epilepsies were genetic, although it was suspected that they were. And it's turned out that many of them in fact are, and that's what I'm going to focus on. And a lot of this is going to be work in progress. There are lots of answers we don't have, but I think it'll illustrate the point of what translational research is all about. So you've heard the term seizures and epilepsy. I thought, since not all of you are epileptologists, I would start out with a few definitions. Here we go. First, what are seizures? They're the clinical manifestations which include symptoms and signs of excessive and hypersynchronous, usually self-limited, abnormal activity of neurons. All of us in this room could have a seizure given the appropriate drug, for example, and the appropriate condition that might render part of our brain non-functional, say with a head injury. That doesn't mean, however, that we have epilepsy. So that's a property of the brain to generate seizures under certain pathological conditions. Epilepsy is a chronic disorder characterized by recurrent, unprovoked seizures. So this is an entity where there is some type of change in the nervous system that will cause these seizures to occur spontaneously. So it's epilepsy that we're focusing on now. There are two kinds of epilepsy we generally talk about. One type is idiopathic, which we now know probably is almost entirely genetic. And the other form are symptomatic or acquired. And you can see all the causes of acquired epilepsies on the right. There are many, many ways of damaging the brain and having it spontaneously degenerate seizures. I'm going to focus today on the idiopathic epilepsies, again, which are primarily genetic. Now, there are a whole ton of these. For the medical students, this is just a partial list of some of the idiopathic epilepsies that have been described. But we generally break them down into three categories, just to make them simple. One are the idiopathic generalized epilepsies. These are epilepsies that occur throughout the brain. They're probably thalamocortical epilepsies. Both thalamus and cortex are involved. Pathways are used that are similar to sleep pathways, involving the thalamus projecting to cortex projecting back to thalamus. Idiopathic focal epilepsies, however, also occur. So genetic focal epilepsies are a reality. There are many fewer of them, but they do occur. And then finally, Epileptic encephalopathies. These are diseases that occur in childhood. They often are heralded by the onset of epilepsy, and they are associated with severe developmental disability and often death. These are very severe, and they're often called catastrophic epileptic encephalopathies. I'm going to focus in this talk on the idiopathic generalized epilepsies and a little bit about the epileptic encephalopathies, and specifically childhood absence epilepsy, childhood epilepsy with febrile seizures plus, and juvenile myoclonic epilepsies. Those three entities have varying severity, and you'll see have a different genetic basis. Childhood absence epilepsy is a disease where children, usually pre-adolescent or peri-adolescent, who develop sudden absence seizures where they blank stare for 10 or 20 seconds and then recover very quickly. It can be debilitating in that there's a lot of lost time. So school performance, for example, can be impaired. Often children grow out of these spells. That is, when they become adult, they don't have seizures. Generalized epilepsy with febrile seizures plus, or GABAA plus, I'll refer to it as, is a more severe condition which has both generalized tonic-clonic seizures, febrile seizures, and afebrile seizures that occur after the age which you'd expect febrile seizures to occur. And there are also myoclonic seizures and other seizures. So if you look at a pedigree of GABAA plus families, they'll have individuals who have very different kinds of epilepsy, but they all have a common genetic basis, and so it's part of the syndrome of GABAA plus, a little worse than childhood absence epilepsy. Juvenile myoclonic epilepsy is a more severe form in the sense that it has generalized tonic-clonic seizures, myoclonic seizures, and about 30 or 40% have also absence seizures. This type of epilepsy you do not outgrow. This is a lifelong epilepsy that requires management and treatment the entire lifespan. And then finally, epileptic encephalopathies. I'll only talk about Dravet syndrome. Dravet syndrome is an extremely serious condition. Children often develop myoclonic jerks, sudden jerks, at very young ages, less than 2, and then have severe developmental delay, often leading to death. So this is, again, a genetic epilepsy, often sporadic, often not inherited, although sometimes. So, IGEs, as I said earlier, were thought to be genetic. It wasn't clear what the genes were. And over the last 15 years, this area has just absolutely exploded. It's quite clear that most of the IGEs are associated with ion channel mutations as well as variants. And they have clear genetic components. The first cloning of epilepsy genes occurred in families that had autosomal dominant inheritance of their epilepsy. And so these fully penetrant monogenic alleles, and there are some that are less penetrant also, only occur in a very small number of patients. About less than 2% of the idiopathic generalized epilepsies are associated with monogenic epilepsies. The vast majority of them are related to rare polygenic alleles of small effect. Most of the talk will be on the former, that is the autosomal dominant rare epilepsies, because we've learned a lot about what these mutations do to the function of the ion channels. But I'll end up with a little bit of sort of work in progress on how we're beginning to approach polygenetic epilepsies and try to understand their basis also. So we'll start with the fully penetrant monogenic alleles. So this is a slide that shows a series of ion channel human epilepsy genes that I'll refer to as HEP genes. I'll try not to, but I always fall into that. HEP genes are genes which have been shown to be directly related to the development of idiopathic epilepsy. So these are all supported by genetic studies that this trait is inherited with the mutation, not present in family members who do not have the mutation. You can see there are ligand-gated ion channels, bullet-gated ion channels, calcium-activated ion channels, as well as chloride ion channels. For this talk, as Ken alluded to, I'm going to focus on GABAA receptors because that's what I do. We have all the reagents to study them, so we focused on this. But everything I'm going to tell you can be generalized to all the ion channel mutations because there's nothing special about this gene family. It just happens to be one that we have studied very intensively. So GABAA receptor genes, there are 19 of them. They have different Greek letters, alpha, beta, gamma, etc. You can see the list there. There are genes that are on different chromosomes. They're individual genes. But the protein products that occur are very, very similar, very conserved for a family. For example, alpha 1, 2, 3, 4, 5, 6. Each one of those peptides are 70 to 80% homologous with each other. Between families, alpha to beta, for example, there's about 20 to 30% sequence homology. So 19 genes, they're all different gene products, different locations on chromosomes. But there are only 3 HEP genes in the GABAA receptor family, at least so far, alpha 1, beta 3, gamma 2. That's a whole discussion we could have, why there is a subset of a gene family that are HEP genes and the other ones may or may not be. We don't know if the other ones are susceptibility genes, and we don't know for sure they're not HEP genes. Time will tell whether or not families are identified that have representative epilepsies associated with these genes. But I'm going to focus on these 3 HEP genes primarily. So what are GABA receptors? Well, this is a GABA receptor subunit just showing the linear sequence and a little bit about the topology. These are cis loop receptors. They have a large extracellular domain, which is shown up here. They have 4 transmembrane domains. The M2 domain, as you'll see in a minute, forms the ion channel because these actually are pentamers. And there's a large cytoplasmic loop between the M3 and the M4 segment, right in here. Now, what they actually look like is this. This is a modeling program that allows us to use known crystallized structure and then infer from it, because the GABA receptor's not actually been crystallized, but cis loop receptors have, and C. elegans or worm forms have, that are very, very similar to these. So this particular peptide has a large internal domain, which is the same thing here. It has 4 alpha helices, shown over here, and then has a large intracellular loop. This structure should be ignored. There's no real known structure of the cytoplasmic loops. It's probably, a lot of it's not coiled or sheeted, it's just plain free. I've already told you about the 19 genes. The 19 genes all have 19 protein products from those genes, and this shows these families of 6 alpha, 3 beta, 3 gamma, etc. Now, these subunits are very well organized. They're actually polar. This particular subunit, and all of them, have a similar plus side, which is this particular side of the molecule, and a minus side, which is this side. I'll show you in a second why that's important. The GABA binding site is made in a pocket right in this location between 2 subunits that come together in an adjacent way, forms the binding pocket for GABA to activate the GABA receptors. These are in fact pentamers. 5 different subunits form a receptor. They're heteropentamers most of the times. There are homopentamers, but most of them are heteropentamers. And this particular construct shows an alpha 1, beta 3, gamma 2 receptor. Stoichiometry is 2 betas, 2 alphas, 1 gamma in general. And the stoichiometry sequence is GBABA, looking counterclockwise as you look at the receptor from the top. So 2 betas, 2 alphas, and a gamma. You notice this beta subunit right here. This is looking down on the receptor. The ion channel, the chloride ion channel is right here. This is the minus side, this is the plus side. This would abut a minus side of alpha to a plus side of alpha, and so around the circle. So all the plus-minus interactions and minus-plus interactions determine the polarity of these subunits. So they can't randomly fit in there. They have to fit in with adjacent subunits, and they have to fit in with a very specific polarity. This will be important as we go along to some of the mutational effects on these subunits. This is the same thing shown here, but now it's a vertical view. Again, red is alpha is red, beta is blue, and the gamma is silver. You can see the transmembrane structures, the alpha helices right there. There's no structure shown here in the cytoplasmic domain because, as I said, there is no structure. It's just basically a free string of amino acids. Okay. Now, all these subunits are formed in a very, very standard way. All the students here know this, the synthetic pathway for these. Everybody knows the secretory pathway, and this is nothing different here. This could be in any cell in the body. It also occurs in brain. 4 main steps I want to emphasize. All these genes have to be transcribed. This occurs in the nucleus. Once the mRNA is produced, it attaches to ribosomes in the ER. Protein is translated. That protein, since this is a highly folded, organized structure, has to fold, be glycosylated, form disulfide bridges. Everything that happens post-translationally occurs in the ER, so these subunits are formed and become immature but folded and ready to assemble subunits. After that, receptor assembly occurs. Receptor assembly requires that these subunits oligomerize, that is, they couple to each other, and obviously you have to form a pentamer. So the thinking is there are probably dimers that form, then the dimers of dimers, and then a pentamer is formed when the 5th subunit comes in and docks. Presumably alpha, beta, and then a gamma comes in and completes the pentamer. This forms an immature, not fully processed receptor, which then has to be trafficked to the Golgi, has to be then reglycosylated with mature glycans, and then is trafficked to the surface membrane. Then the final step is it reaches the surface membrane via these endosomes. There's exocytosis, at least fusion of the membrane, and now you have a fully functional receptor that's sitting and targeted to the right location in the nervous system, either a postsynaptic inhibitory location or a extrasynaptic location that may mediate a different form of inhibition. So 4 basic processes for synthesis of these receptors. Now, getting back to the human epilepsy genes, these genetic epilepsies have been associated with about 20 mutations. and counting. These occur quite frequently, and I'm sure we'll end up with many, many more. The epilepsy mutations themselves all have slightly different effects, but I'm going to show you what the general effects are, and then we're going to go through a classification of these mutations to see where we're going and trying to understand the pathogenesis of these epilepsy mutations. So they've been shown to impair gene transcription, primarily by putting mutations in the promoter region, which drives transcription. Subunit biogenesis is affected either by processes that degrade the subunit message, mRNA, or degrade the protein itself. Receptor assembly trafficking is disturbed, causing ER retention, that is, these immature forms of the receptor can't get out, they retain in the ER, due to either subunit truncation with a dominant negative effect, that is, a truncated protein which sits in the ER, oligomerizes with good subunits, but prevents them both from getting to the surface. So there's a dominant negative effect. Or impaired subunit oligomerization, so normal subunits that have a decreased affinity to oligomerize, and so they don't efficiently produce receptors. Then finally, receptor channel function, that is, the receptor is formed, oligomerizes, traffics to the surface, but once there, doesn't act normally. It has impaired function. So we're interested in why genetic epilepsies occur, but we're also interested in fixing them. There is no surgical approach to treating these. It's going to have to be a pharmacological or an interventional approach, and there are a variety of things I'm going to share with you. But you can't design a therapy for every single boutique mutation that occurs that may occur in 3 families or one member of each one of 3 families. So our strategy has been to try to classify these mutations into subgroups, all of which have the same basic pathological mechanism, so we can target therapies to a class of mutations. And again, this would be true for other ion channels also. So I'm going to take you through the 6 different classifications and show you a few examples of what they do. So here's our cell biology secretory pathway shown again. In the nucleus, there are a promoter mutation. There's one in the beta-3 subunit. It causes reduced subunits due just to impaired transcription. You don't get enough message. You don't have enough message. You don't get enough protein. That causes decreased beta-3 subunits, which causes decreased inhibition in places where beta-3 subunits are important. Second is a biogenesis defect. This is in GABAA receptors when you have a nonsense mutation. Nonsense mutations are mutations that cause premature translation termination codons, or premature stop codons. You get a translation that goes along, hits one of these, stops translation, now you have a truncated protein. Why is that important? Well, it's important because, again, you can have dominant negative effects. You have these truncated proteins that sit in the cell, they oligomerize with good subunits, and now you can't get subunits to the surface because these short truncated proteins are preventing them from trafficking. The GABAA receptor is a multi-exon gene. There are usually 9 exons. And if you have a mutation that is not in the last exon, and I'll show you a cartoon in just a moment, and in the penultimate exon, 50 to 55 nucleotides from the last exon-exon junction, you will find that the mRNA is degraded, and that's what happens with these particular ones. You can see that the mutations are in gamma-2, there it is, gamma-2, and alpha-1, and they're generally associated with childhood absence epilepsy. You'll see that in a moment. Not a very severe form. This is what they would produce. These are again, using the modeling program, using known sequence to predict what the peptides would look like inside the cells. And you can see they're all aberrant. They're truncated, shorter or longer, depending on where the PTC exists. These would all produce dominant negative effects, especially the 2 on the right. The 2 on the left might not, because they don't have enough of this internal structure to actually oligomerize with normal subunits. But the 2 on the right certainly would. However, it's very unlikely that'll happen because of ER quality control. This process of nonsense-mediated mRNA decay is activated because these mutations are all in early exons. I didn't do that. Okay. So this is a cartoon mRNA, and it shows early exons on the left, well, my left, your left. Shows an exon-exon junction, and then it shows the last exon on the right. The idea is if the PTC is in an early exon and there's an exon-exon junction, 50 to 55 nucleotides at least from the mutation, then when the ribosome hits the PTC, it will initiate nonsense-mediated decay, which will activate a process for degrading the mRNA and making it go away. This occurs on the pioneering round of translation. So there's a little protein product that's produced, but not very much. So if NMD is activated, it will basically cause a haploinsufficient state, You lose the product of one allele. And you'll have disinhibition because there's just less mRNA. Third class are missense mutations, not nonsense. These are missense mutations that change the amino acid identity, but don't cause a premature translation termination codon. The one shown here is in an alpha-1 subunit. It causes a subunit to be misfolded and then ultimately degraded. This particular subunit is in the third transmembrane domain. It's a highly non-conserved substitution, D for A. So you have a highly charged residue being substituted for a non-charged small residue. We've shown that, in fact, what it does is it causes misfolding of that M3. The third transmembrane alpha helix can't insert in the membrane, and so it's on the wrong side of the cell. It's outside of the extracellular space. This is a big flag, so it can't assemble. And so what happens is it's recognized as an unfolded protein. Again, ER quality control jumps in, and by a process called ER-associated degradation, or ERAD, this protein is degraded. What happens is there's a timing sequence inside the ER that's looking for folded proteins to complete their folding. If they can't fold in time, they are then shuttled out, dislocated to the cytosol, which is shown in here, polyubiquitinated, and then shuttled to the proteasome. The proteasome then degrades them. And so it's a rapid way of eliminating misfolded proteins. Fourth class are mutations that are in the last exon. I made a big deal about NMD when you have an early exon PTC. If you have a stop codon that occurs in the last exon, NMD is not activated because there's no exon-exon junction downstream from it. So what that means is, here are again modeled subunits from 4 different mutations, all associated with PTCs in the last exon. You can see they look very much like receptors. They're all missing transmembrane 4. The truncations are all somewhere in the M3, M4 loop, or there's a frameshift mutation that causes extension. That red is nonsense sequence that was initiated simply because it was a frame shift in the protein. All of these subunits have all of the extracellular domain intact, so they would all oligomerize very effectively. We've shown that all of these have very strong dominant negative effects. And you'll notice that the epilepsies they're associated with now, instead of being childhood absence epilepsy and febrile seizures, they're now all GEFS plus or generalized tonic-clonic seizures. So more severe biology, more severe pathology, more severe epilepsy. So that makes sense. Class 5 mutations are missense mutations. These mutations are mutations that cause impaired oligomerization of subunits. The subunits can come together, they can form receptors, but they don't do it as efficiently as wild type or normal receptors do. There are a whole bunch of them, and you can see them all listed in gamma and in beta. They're again associated with less severe epilepsies, childhood absence epilepsy and febrile seizures primarily. This is where they're located. This is in the N-terminus. The red box is a whole series of these mutations. They're all in similar locations that I'll show in just a second. They're in the extracellular domain, and it turns out that they're in interface domains where subunits oligomerize. This is a real busy slide. Let me just take you through it. So what we've done is to include the actual amino acid changes on the structure of these subunits after they've been assembled. So what you can see, and the easiest one to look at is this horizontal section right there. This is now exploded here. And you can see N79S, P83S, G32, which is in beta 3, Q82, R82Q, and R177G. These all live at the gamma plus beta 3 minus interface or the alpha plus gamma 2 minus interface. Remember I said those interfaces were very polar. Okay? And so what's happening is when assembly tries to occur, you've got a mutation right at one of the interacting sites, it decreases the efficiency of oligomerization. So it decreases the efficiency of making receptors. So it decreases the efficiency of putting them on the surface. But once they get to the surface, they're normal. They're perfectly normal. And that will be important from us, a therapeutic strategy in a moment. So all these seem to have a very common mechanism and produce similar epilepsies. Then the last one, class 6, this is another missense mutation. These are in gamma 2 and in delta. The delta is actually a variant, not a mutation. It's seen in the general population, not very often, but it's associated in families that have, in this particular case, GEFS plus epilepsy. These are mutations that don't interfere with assembly, don't interfere with trafficking, don't interfere with insertion in the surface. But once they get there, they misfunction. And what they generally do is the channels open and close, they're gated. And you see a very characteristic gating pattern for a normal GABA receptor. When you have one of these mutations, the gate opens, current flows, but it immediately shuts. So you get very brief openings. Those very brief openings translate into decreased current, which translates into decreased inhibition. Okay. These mutations, in contrast to the earlier ones, which were all located in the extracellular domain at binding sites, these are all in the transduction domain. These are all in locations very near the membrane, where the GABA binding to the receptor causes conformational changes, causes the gate to open. And these mutations are sitting right in that transmission, that transduction zone that couples binding to gating. So it fits geographically. Okay, so what? I've just given you a big laundry list, and I could have shown you all the basic science behind it, but we'd be here for several hours, so just hope you'll accept the fact that what I told you is true. Why do we do it? Well, we did it to develop therapeutic strategies. You can't develop rational therapies unless you know the basic pathophysiology of the disease you're trying to treat. You can't just take pharmaceuticals, although most of the drugs we use in epilepsy were serendipitous in terms of their discovery. That's not the way things are done these days. It's certainly not rational. So what I'm going to go through is those 6 classes of pathology and propose 6 ways of treating them. Some of them make sense, some of them don't make sense. And let me make another point. You would not develop an extremely dangerous strategy to treat a patient with childhood absence epilepsy. They'll likely outgrow it. We have pretty good medicines for it. That's not a category. On the other hand, somebody with Dravet syndrome, you would treat. You would go to extreme lengths because these patients are going to die. These children are likely going to die. So they're individuals you would treat. Someone with a lifelong epilepsy, if you're a parent of a child who's having seizures, once a week or once every 2 weeks or twice a day, you're going to be willing to go to some more aggressive treatments to stop those seizures. Okay, so it all depends on the severity of the epilepsy, and I'm not going to pay attention to that. I'm just going to talk about the general strategy. So class 1 therapy. These are reduced subunit due to impaired transcription. The best way you could treat this would be to develop transcriptional activators that would in fact overcome the transcriptional block due to the mutation in the promoter. Second one, class 2 therapy. These are reduced subunits due to impaired translation. These are the ones where NMD blocks them. To do this has been done in cystic fibrosis. You can use a drug that would be better than, but similar to aminoglycoside antibiotics. These glycoside antibiotics bind to ribosomes, and during translation cause read-through of premature stop codons. And so they produce full-length peptides. They don't stop recognition of the last stop codon. So what that does is it actually, if you could get enough protein, would cure the disease. Because once you read through it, you have a normal peptide. Okay, we've shown in the dish that we can in fact do that. You don't get a large amount of rescue, but with aminoglycosides, you do get some. There are a number of pharmaceutical firms who are trying to develop better compounds that will cause better read-through. But this would be an extremely exciting strategy for cystic fibrosis or for genetic epilepsies. Class 3 therapy, subunit misfolded and degraded by ERAD. Here you'd have to upregulate the wild type or replace the mutant subunits, and that may be getting into what I was calling more dangerous therapy, gene therapy in the future. This assumes that better techniques will be developed to deliver genes to the nervous system in a more targeted fashion. We're trying to do this. We don't have a gene therapy that would be appropriate in humans. What we're doing is a genetic approach. So we've created a back mouse, a mouse that carries a transcription of a wild type gamma-2 subunit, human gamma-2 subunit, and we're crossing it with animals that have mutations that would be therapeutically appropriate, and seeing by introducing the transgene if we can cure the disease. And so far, in the preliminary days, it looks like we can. So it's proof of principle that if you can supply using some technique an exogenous gamma subunit, you can overcome the disease by replacing the lost subunit. Class 4 therapy. Subunits are truncated with or without dominant negative effect. Again, using an aminoglycoside antibiotic or something to read through, or to knock down the dominant negative gene. You've got another problem with these, because it's not simply replacing the subunit. You've got to deal with the fact that there's a dominant negative gene in there. And it's not clear that simply providing more wild-type gamma subunits will cure your disease. It may be okay, but not totally clear. So you may have to also knock down the dominant negative gene. Class 5 therapy, ER retention with impaired subunit oligomerization. These, again, are these ones where the subunits will oligomerize, they just don't do it efficiently. And so the best way to do that is to have what are called correctors or pharmacological chaperones that will cause an accelerated interaction. And once you accelerate the interaction, the receptors you form are normal. And so you don't have a problem having to provide external subunits. You just have to improve the efficiency of assembly and trafficking of the ones you've already got. Then finally, class 6 therapy, use specific or non-specific GABAA receptor positive allosteric modulators to enhance the current. They're called channel potentiators. We have a number of them, benzodiazepines, for example, barbiturates, neurosteroids, but none of them have been shown to effectively treat these kinds of epilepsies. And there are lots of pharmacological reasons why, which I'm glad to deal with if someone wants to ask me a question about it. Okay, so those are the monogenic epilepsies. Now, I want to spend a few minutes on a more pressing issue, and that is the polygenic epilepsies, for which we've made absolutely no progress at all. This is an extremely tough nut to crack, and I just want to show you the initial stages of what we're thinking about trying to approach these. So here's the problem. Everyone in this room, if we were to sequence your genome, you'd have 3 to 4 million genomic variants. Huge number of variation in your own genome. You can break this into rare or common variants, and arbitrarily defined as less than 5% of the population. So if you focus on the rare variants, these can occur in non-coding or coding sequences. Well, obviously, for functional effects, you want to focus on coding sequences, not non-coding sequences. And in the coding sequences, you want to focus on non-synonymous, not synonymous. So synonymous mutations are those that change nucleotide but do not change amino acid, whereas non-synonymous change nucleotide and change amino acid. So you really want to focus on non-synonymous mutations, the cause of missense mutation, if you will. Also, if they happen to produce a nonsense mutation, a splice site or a frameshift mutation. Any of those will cause severe alteration of function of the peptide. So, the first steps toward doing this was done by Tara Clausen and Jeff Nobles at Baylor. They did a large collaborative study using whole exome sequencing. So they identified 237 excitability genes, calcium channels, GABAA receptors, every excitable channel you can think of. And they did whole exome sequencing through those genes. That means they got the open reading frame, they got the introns, they got some 5' sequence, including the promoter, they got 3' downstream sequence also. They just did bulk sequencing, and then they looked to see what they got. And they had two populations, one, patients with IgEs, and controls. Now, there's no family information here. There are no kindreds here. These are just identified patients that walk through the door that have a generalized epilepsy, and they get sequenced. So you don't know anything about inheritance of any of these variants. So what did they find? Well, they found non-synonymous, some nonsense and splice site SNPs, single nucleotide polymorphisms. But by far the non-synonymous SNPs were the ones that were most important. 668 of them out of 3,000 total SNPs, so about 20-ish%. So that's interesting. So the idea here is maybe you've got non-synonymous SNPs in controls, non-synonymous SNPs in patients with IgEs. We ought to be able to segregate them, separate them, look at the SNP patterns, and we can say, you have epilepsy because you have this SNP pattern, you don't because you don't. Pretty simple. A little disheartening. What they found was there were lots of non-synonymous SNPs in cases, but there were lots in controls also. And there were lots of non-synonymous SNPs that were shared between controls and patients. And in fact, when they did individual, what they call chanotypes, that is to look at all the non-synonymous SNPs, if you looked at the pattern, you couldn't distinguish controls from epilepsy patients. There was absolutely no information based on SNP burden. And Jeff Nobles, when you hear him talk about it, he'll kind of go, because the good idea didn't work. So right away, the obvious conclusion, you can simply genotype somebody and identify a SNP pattern is probably not going to be true. There has to be something more about it. And I'll show you what we're trying to do very, very early days. We're just beginning to think about this. So the issue here is you can't take bulk SNP burden. You now have to do some filtering. You have to say something about the relevance of the SNPs. And then, as I'll say at the end of my last slide, then something interpreting how you deal with that relevance. So you want to deal with non-synonymous, nonsense, splice site frameshift mutations that are likely to alter function. You want to find absent or rare SNPs, not common ones. If you've got any family information, that would be great, because then you can look and see if there is a chanotype that segregates. For example, a combination of alpha-1, beta-3, alpha-5 SNPs may segregate the disease, and you can now pattern recognize using informatic approaches. That'd be great. Not likely, but it would be great. You can use bioinformatics to predict whether or not the SNPs are likely to disturb function. And I'll show you the example of how you can do this. These are looking primarily at things like You put a charged residue replacing a non-charged residue, that's going to change structure. But these are all done outside the structure of the protein you're studying. They're simply done theoretically for a generic polypeptide. The next you could do in vitro characterization, which I'll show you. And then finally, if there's any gene information available from any database, you can obviously use that. But for most of these SNPs, there's not going to be much information, if any information available at all. Okay, so we took all the exome sequencing from the Nobles group, from Tara Clausen. We pulled out all the GABA receptor genes, and we found in 42 controls and 144 cases, 147 GABA SNPs, non-synonymous SNPs, and 525 in the cases. Because there were different numbers of cases and controls, if you normalize for the number of subjects, it was 3.5 versus 3.6 non-synonymous SNPs per individual. So as they showed with the bulk data, we showed the same thing. There's no difference between the controls and the epilepsy patients. Second, we thought, well, maybe there's a different type of SNP. So the 147 in controls were 15 different non-synonymous SNPs, whereas the ones in the cases were 36 different non-synonymous SNPs. But if you look at the right, you'll see the case incidence is identical. There's no difference between the controls and the patients with epilepsy. So that didn't help. The third thing did help. We now looked at rare versus common SNPs. If you now look at controls, only 13% of them were rare non-synonymous SNPs. But if you looked at the cases, 67% were in fact rare. So that suggests that if you look at the identity of specific rare non-synonymous SNPs as a filtering function, as I showed you earlier in the flow diagram, you've got a chance to find enrichment of a population of SNPs that may code for pathogenesis of these epilepsies. Only a couple of slides left. Here's the list that we found. On the left are the controls only. There are only 2 of them. We're finding controls but not in cases. But there are 24 shown on the right in cases but not in controls. The ones in the middle are novel. They have not previously been reported in public databases. The ones on the right had been reported in public databases. They're still rare, but they were identified and available through the public databases. So what do you do with this list of stuff? Well, we noticed that there were 4 non-synonymous SNPs in the alpha-5 subunit. 2 of them novel, 2 of them not novel. So we thought we'd use those 4 as sort of an exemplar to see if you can make any sense using informatic approaches or using biological approaches. So we used those informatics tools, SIFT and PolyFin, to identify whether they were predicted to produce damaging or non-damaging mutations. The red box ones are novel cases that were predicted to produce altered function. The ones on the right, the blue ones, were not novel and were not predicted. So this is kind of cool. We can look at the novel SNPs, SIFT or PolyFin will predict they'll be damaging. Those must be the real deal. And the blue ones are not novel, aren't predicted to be destructive. They should be the not real deal. Easily testable. This is where these mutations are. You can see they're in vastly different locations. The alpha-5 V204I is in the extracellular domain in a very suspicious location there. The 2 other ones, P45, P453L and W280R, are in transmembrane domains, and the S402A is in the cytoplasmic domain. So 4 totally different locations. And again, differences in prediction. So we did transfected HEC293 cells with these, did whole cell recording just to determine with alpha, beta, gamma constructs whether there was an alteration in current. And in fact, you can see there was. Starting over on the right here, the current density from cells transfected with I've lost the mouse. Oh. You can see that V204I and 280R both have depressed current. Also, from the current traces, you can see there's a different rate of decay, suggesting that the function of those receptors has been altered. The only problem is that doesn't correlate with our prediction, right? It's one of each. It's one of the non-novel, one of the novel, one of the not predicted to be destructive, one of the predicted to be destructive. So it doesn't fit at all. We did it in flow cytometry to detect surface expression of these receptors, and we found exactly the same result. The 2 that had reduced current had reduced surface expression, and the 2 that didn't. So what that says is that using simple informatic tools to predict the function of these mutations is probably not going to work. You have to do functional characterization. But the good news is we were able to select only 2 mutations on those 4 and decrease by 50% the number we had to worry about, because the other 2 were perfectly normal. So that's sort of an insight into our strategy. Last slide. So what are the next steps? Well, I think the next step is to characterize the significance of all the rare variants. Now, this is not a Manhattan project, but it does require some NIH funding. It's simply labs that will commit to studying their favorite protein and determine which ones are in fact functionally significant. That will reduce the big SNP database to a much smaller SNP database. It will help informatic identification of significant genes that may contribute. So the functional chanotypes for controls and cases, I think, may work if we now have functional chanotypes, not simply non-synonymous SNPs. We also want to compare HEP genes to non-HEP genes, because as I told you earlier, it's not clear that non-HEP genes really contribute. So it may be that only HEP genes contribute. That would further narrow the number of non-synonymous SNPs we have to worry about. Then finally, functional chanotype IgE concordance can we pattern identify simply by looking at all those patients that have the epilepsy and those that don't and say, aha, you always see an alpha 5, beta 3, gamma 2 pattern in all the patients, but you don't in the non-patients. And then finally, if we can get some inheritance data, even though these are not autosomally dominantly inherited, we may be able through several generations to identify a pattern of SNPs that is maintained and correlated with the epilepsy. So I think these are very hopeful signs that we'll be able to make a dent in this huge area of polygenetic epilepsies. I'm very enthusiastic, and NIH thinks it's a good idea, so as long as that's true, we'll keep going. So thank you very much. To answer any questions, I put a list up here of folks that did a lot of the work, and I didn't really talk about their specific work, but who did a lot of work in characterizing the pathophysiology of the ion channel variants and mutations.
Just physiologically, when you have these receptor abnormalities, you do electrode monitoring these neurons that are extremely active.
Most of what we're doing are in HEC293 cells, so they're not active at all. They're just expressing the protein. So we can look at the current amplitude, we can look at its kinetics. To get to mice, or to get to neurons, we have to transfect in subunits on top of a background set of mouse genes. So it makes it very difficult to do network studies. So the way we're approaching that is we're taking exemplars. I don't have time to talk about it, but we're making mutations in mice, so making knock-in mice that have class 1, class 2, class 4. And so far we have 3 of the 6 classes with transgenic mice, with knock-in mice. And so now the issue is doing slice physiology and thalamocortical preparations and identifying what the defect is. We're very optimistic that by using the class approach, we'll be able to say, you know, class 5 mutation does this to excitability. And that would answer your question, but we're a bit away from having all classes represented. Yes?
And I suggest that you say your name and who you are, so other people can get to know other people in the room.
It's kind of a big deal. Submit a written question now.
So I'm Gogi Kumar, I'm a child neurologist here. So, you know, for the idiopathic generalized epilepsies, we never do any gene testing. I mean, could you comment on which subgroup should we test, you know, for genetic mutations, and how do we do that? Like which labs test for
Yeah, so I'm not very optimistic about doing much gene testing in very many of these patients right now. The big danger is, let me go back to the Klaas and Noble study. They found a mutation in sodium channels that was a channel destructive mutation, that it caused loss of function. It was the only nonsense mutation they found that caused loss of function, and it was in a control. And so the problem is that a lot of these do not have tight genetic correlation. So many of the mutations that you get coming back, I don't know how you interpret them. So I think the only ones that you want to identify are ones that have very strong genetics behind them, and then only interpret when you find families that have those particular mutations. You know, you can use Athena, or, you know, that's probably the best, largest coverage that you can get. But I think it's very dangerous simply to get the sequence, come up with a SNP and say, aha, your epilepsy is because you have this particular SNP. Because that particular SNP may have absolutely nothing to do with your epilepsy, even though it's in a HEP gene. Yes?
I'm very good I'm an epileptologist. I noticed with some of your sodium channel abnormalities that were present from the epilepsies that they're the same sodium channel abnormalities that are seen in hemiplegic migraine. Can you draw any correlations between the etiology or pathogenesis?
So yeah, so the big issue is when you go from the cell biology of the individual ion channel mutations, you understand the cell biology. What you don't understand is the role it plays in the network. Epilepsy is a network disease. It's not an ion channel disease. It's not a neural cell disease. It's a network disease. And so the mutation that you have depends on what cell population it occurs and what that network is. And so whether it's paroxysmal dyskinesias, or hemiplegic migraine, or epilepsy of different types, they all may have similar mutations in similar genes, but have different clinical manifestations because they have different background genes and they're affecting different networks.
Well, I've always thought that hemiplegic migraine is a neuronal dysfunction rather than vascular dysfunction.
Right, I agree. But the network in which it's embedded is extremely important for the things we see, the symptomatology and obviously the therapy we're trying to treat. Yes.
I'm Francisco Alvarez-Nicholas from the Department of Pharmacology. I would like to ask you, apparently your mutations are in different locations in the predicted three-dimensional structure of the GABAA channels. And what you show there in the hex cell experiments is that the currents actually get shorter or lengthened with respect to the wild type. What do you think is happening? Is it a problem in the channel gating, or is it a problem in the affinity for GABA for the receptor?
So we don't, in those particular ones we don't know because we haven't done single channel recording. If we were to do single channel recording, we can tell whether the kinetics are normal or abnormal. So I can't answer the question. I could say in general, most of the GABA receptors we study using kinetic approaches, they're not affinity problems per se, they're actually problems with gating. And so you have different amounts of desensitization, for example, which influences the kinetics tremendously, different stability of open channels. So you have to characterize all those characteristics, and then you can figure out why the channel, why the kinetics are altered. Yes?
Sort of related and introduced already from the group. Sort of related question in your expression systems. Do any of the allosteric modulators work even on the mutated channel, especially if the mutation is indicating?
So pharmacological question. So the question here is, with the mutant receptor channels, do allosteric modulators like benzodiazepines or barbiturates and neurosteroids still work? And the answer is you have to test each one. Benzodiazepines, for example, act at a different interface than the GABA binding site. And so depending on where the mutation is, you may expect or not expect there would be an effect on an allosteric modulator. However, you don't know if there's an additional disruption of inter subunit binding because there's one interface that's abnormal. It may propagate to another interface. So that has to be tested for each of the different constructs. So it's a very good question. And it's obviously very important for the whole idea of class 6 mutations. Most of those drugs are going to be potentiators. And so you're going to want to know that they bind and in fact will increase trafficking.